Hi everyone. Welcome to those who are new to Malaysian Research Insider. And to those who have joined us last Saturday, welcome back. I'm Apple. I'm doing my PhD in molecular plant science at the University of Edinburgh in the UK. And this science chat is co-organized by MBIOS, 100 Scientists of Malaysia, and our media partner, Science Media Centre Malaysia. So let's welcome our scientist today, Sabrina. She's in University of Adelaide doing a PhD in Plant Epigenetics and Bioinformatics. So over to you, Sabrina.
All right, I'll start sharing my screen. All right, so thank you so much, Apple, for introducing me today. It's always honored to have another plant scientist introducing a plant scientist, and I was actually pretty stoked to actually find out she'll be hosting my session today. So the title of my today's talk is Hearing Whispers, Deciphering RNA Modification in Plants. I'm Pei Qin Sabrina Ng, currently a third-year PhD student at the University of Adelaide. And before I start my talk, I would like to thank to the organizers, so MBIOS and 100 Scientists of Malaysia, for actually contacting me and giving me this wonderful opportunity to speak about my work, as well as our media partner here, Science Media Centre Malaysia, and for those of you who actually take time and listen to my talk today. And so to start off my talk today, I'll briefly introduce myself. So some facts about me, how am I like when I'm in the lab and when I'm outside my lab. Then I'll go into the next topic, which is looking at language in a different world and its RNA modifications. It might sound unfamiliar to you, but don't worry, we'll slowly look into it and see what RNA modification actually is. And the key point of my talk today is actually about things that we have been posting on Facebook so far, if you did see the Facebook post. So what are plant cells actually whispering about? So do they actually whisper? And if they do, what do they actually talk about? And can we understand them? Finally, I would like you to take away the significance of understanding the whispered messages and understanding how and why is it important for us to understand these messages that might seem foreign to us, but at the same time familiar. So about me. So this is me in the lab. So I have two identities. Yes, I'm a PhD in plant epigenetics. That means I do wet lab, lots of experiments, hopefully not exploding things in the lab. And I've also working on bioinformatics. So probably a more common way to say it is, yes, I do code, I smash the keyboards, I type nerdy computer codes to try and solve big data problems. And I do both of them in my PhD research. And the title of my PhD research is, that's for my thesis, is called The Role of RNA Modifications in RNA Processing in Arabidopsis thaliana. Yes, very mouthful, very scientific, but in a simpler way of saying it is understanding the language that affects passing on messages in a plant called Arabidopsis. So, well, two different images. Basically, I'm looking at the same thing. So semi in the lab kind of identity, I do a little bit of science communication, and I do occasionally write blogs as well. So I run a blog called The Data Miner's Diary, where I document my daily lives as a PhD student, previously MPhil, now PhD, my research journey and my thoughts about it. The main motivation for me to start this blog is because I find that the resources that's available out there is fairly limited for PhD students or people who actually think of venturing into research. And I hope by sharing my experience, both positive and negative ones, hopefully that kind of reflects what actually happens when you're part of the research field, and that could also shed some positive light into how you should look at things, whether if it's the positive aspect of it or negative. Besides that, this year I've been quite active in science communication, probably due to the lockdown or probably due to my recent activities on Twitter. So yes, I do have a Twitter account if you're interested to follow me and hear me talk about my science, my daily events, about my work and my life. So recently, I've actually written a couple of blog posts for different platforms. One of them is Editage Insights. I've also got invited as one of the panelists to talk to scientists about the importance of science communication, as well as being interviewed for my PhD experience by various different platforms. So that's a little bit of me about my science communication aspects. And what about me outside the lab? Well, I'm not always that nerdy, and it doesn't mean that I do science all the time when I'm not in the lab. So outside the lab, I take my coffee a little bit too seriously. So I like to actually like figure out the proper ways to brew coffee. I do attend barista classes. I appreciate nice latte art. Yep, that's the form. That's the kind of art that I do actually appreciate. I do enjoy baking, and recently I'm into like floral piping. As you can see here, like these are some beautiful cupcakes that I made, like, you know, as part of my leisure hobby. Well, other things that I do enjoy doing, including bouldering. So that's a new sport that I kind of got into, and I really find it quite relaxing isn't a word, but I find it quite physically and mentally challenging, but it does take my mind off work for a little bit. Not this year, unfortunately, but usually I do enjoy like occasional traveling, and these are just some of the photos that I took while I was traveling in the past few years. So yeah, that shows a little bit of the adventure side and the outgoing side of me, besides sitting in front of a computer typing code or just at the bench running experiments. Okay, I think that's enough of an introduction about me, and let's get into our topic today. Do plant cells really talk? Before, to start off this topic, I would like to introduce you to the model plant organism that I use in my research. So this is Arabidopsis thaliana. So it's a small plant that looks like this, and then these are the calyx, or where the seeds are. This is how the pollen grain actually looks like. There's a beautiful little flower here, and this is the mature structure or the mature plant. So you might be wondering, there are so many plants out there in the world, and why do we actually choose Arabidopsis as the model plant or as the first step of understanding plant genetics or biology? That's compared to other relatives of Arabidopsis, which means they are actually part of the same family. So what plants are actually in the same family as Arabidopsis? You have your broccoli, Chinese cabbage, canola, and also cabbage. So you might be asking, why can't we just conduct research on crops such as these to actually help us understand plant genetics, rather we pick something that's rather unusual? There are multiple factors to this, so I'll pick the most straightforward one to demonstrate why Arabidopsis is the go-to plant before we kind of expand our research onto like these other crop plants. So your broccoli takes about 20 weeks to reach mature stage. That means ready to harvest and look like the ones that you see in the market. For your Chinese cabbage, it takes about 12 weeks from germination to maturation. For your canola plant, 15 weeks, not too long, but they can get really big. So probably as tall as I am when they are at a mature stage, makes it really difficult to work with them if you are thinking of running experiments on them. And for cabbage, it takes about 25 weeks. Not the easiest plant to harvest, probably not the easiest plant to work with in terms of genetics as well, especially if you try to get any molecular materials out of it. While for Arabidopsis, it's almost like the size of my hand here, and it could grow slightly bigger just than my hand. So it's easily grown, it's manageable, it is small, and since it's the close relative to all these crops, why not use them? And it only takes 8 weeks to reach maturity, and you can start experimenting on them way before their mature stage so that you can actually get useful molecular information out of it. And this started all the way back in the 1950s. So it has a long history to it, and we have a lot of genetic resources to help us understand about plant genetics. And hence, this is why this is the choice of a model organism for plant genetic study. So you have your Arabidopsis here, or let's say this is a representation of a plant. Inside the plant, you get millions and millions of these kind of structures known as plant cells. So this is just a structural representation of one of them. Inside the plant cell, let's see how messages are being encoded, transcribed, and translated. So messages are kept in the database known as DNA. As you can see here, that's the first structure, the double helix structure that you see here, which is colored in blue, red, and orange. And then the message gets copied out into a temporary format known as RNA. And some of these RNAs actually get translated based on its content into its respective proteins. Sounds really simple, right? Just 3 steps. But is that really it? So now we'll revisit this figure that I've shown you earlier, and I'll paint not a full picture, but a much more detailed picture of what's actually happening inside a cell. We'll focus on this part of the process. So from the RNA, maybe to protein, maybe not. An RNA, like I said earlier, is a message. So a message in our world consists of alphabets. Alphabets made up words, and then words made up sentences. That's the same in the cell world as well. But instead of having like the 26 alphabet system, I hope I get that right. So instead of having like multiple alphabet system, there are only 4 base alphabets known as adenine, guanine, cytosine, and uracil. So A, G, C, U. I would like to point out to you that these are all represented by a single alphabet in our field of study as well. So if you kind of have a string of these alphabets, so that I call them, you actually have a string of sentence in the cellular world. And does this string of alphabets, like comprising of adenine, guanine, cytosine, and uracil, does make sense? Like for our sentences in English, a plain sentence might mean something, but it might not be in the right context. The same thing goes with RNA as well. To make sure that the messages mean what they actually meant, they need punctuations, just like English. But the punctuations on RNA are known as RNA modifications, represented by these blinks that I've actually drawn on this stretch of RNA right here. So what are these blinks or these declarations of RNA, or so-called punctuations, actually are? So what happens to the base or the 4 alphabets that I've shown you previously? They are actually being modified by adding different groups onto it. And the most common ones that are being added to these 4 bases or alphabets are actually the methyl group. That, as you can see in all these 4 structures here, the CH3 group is the methyl group that I'm talking about, which is represented by the diamonds here. So these methyl groups that were added on these individual different bases actually acts like a punctuation that gives the context to this message, so that the right message that's being transcribed can get across and then being executed as desired. So how many punctuations are there in the cellular system? For us, we have comma, exclamation mark, full stop, etc. But so far, since the 1980s, this field of study has started, we have identified more than 150 different punctuations or RNA modifications in the cellular world. Some of them are known to have severe effects on growth and development. Some of them, we still don't know what they are actually doing yet. And most of these modifications are currently studied individually. So maybe the methylations of cytosine is being studied as a single case, the methylated guanosine is studied as another case, but we haven't actually got a chance to look at the full sentence. That means looking at multiple punctuations / RNA modifications together to try and piece it together, how these things actually comes into a proper sentence. So up to this stage, I've represented the single string of RNA as sentence or plain sentence, the blinks or the diamonds as punctuations. And why is it important for us to understand these whispers? The reason why I call it whispers is we can't hear them, because if people are talking, you can actually hear what they're actually talking about, you can hear voices, but these are actually ongoing without us actually hearing it. But why is it still a matter for us to understand and research on it? So the classic example of the importance of punctuations, as represented by this meme here, is with one comma missing, you get a complete different meaning of the same sentence. It might actually mean a friendly invitation that you're inviting grandma over for, a nice dinner or lunch, or it might simply just mean you're having grandma for dinner. That's really bad. And that's only just one punctuation difference. So if you're putting it in the RNA context, having this single modification difference could actually change the entire context of the RNA as well. And hence, this is why it is important for us to understand when cells are writing messages in this way, what they actually mean. Are they trying to tell the other components of the cell something that they are concerned about, or that they wanted actions to be taken? So now with this analogy being explained, what we are talking about modified RNAs is actually sentences plus punctuations. As such, that gives it context. So to help us understand like sentences, we need a dictionary, just like how we used to learn other languages. So let's see how far are we in terms of constructing this dictionary to understand RNA modifications. Not very much actually, despite the research actually started way back in the 1980s. This is because of like several factors, for example, technology limitations, and also it kind of progresses together with the genetic research as well. So people tend to start focusing more on proteins and genetic research, thinking that most of the genome is actually, that doesn't encode for protein, is actually junk. But what researchers soon found out that these part of the genomes that don't encode for protein were actually encoding informations that help other processes, such as messages, transferring messages, encoding messages like RNAs, to facilitate the messages to be conveyed as accurate as possible, but not necessarily ending up as a product or protein. So in our lab, what we actually get, so now we have actually described mRNA as the message, and now I would like to introduce you to the messenger, tRNA. So at the early stage of research, our lab actually first studied modifications that's related to cytosine, or represented by alphabet C. And what the lab has discovered was on mRNAs and tRNAs, which is on the message and the messenger itself, they both contain of methylated cytosine. So that's the first type of modification or punctuations that's being found on these two types of different message and messenger. So that kind of suggests that it's not just the message that's kind of carrying these punctuation marks. The messenger itself does have to have the same punctuation mark to enable effective communication between these two. What's more interesting is when the plants lose the modification here, as shown in the animation. So if you lose the punctuation or the modification of a methylated group, the plants actually do have a shorter root phenotype, as shown here in this figure, which to us is actually very surprising. This shows that this modification, to a certain extent, does communicate to the plants, telling them that, okay, I need you to grow roots at this length to facilitate growth. But when you lose this modification, that means you're losing that comma maybe, or that certain punctuation mark in the sentence. It just go, I know you're trying to say something about roots, but I'm not quite sure what you meant, but I'll just make roots anyway. So that kind of shows as an effect of a shorter root phenotype. And to us, that's totally surprising. But what is also surprising is that losing that methylation mark, or losing that modification, actually causes the plants to be more sensitive to oxidative stress. Okay, At this point, you might think, what's actually oxidative stress to plants? But if I said it in the context of free radicals, you might understand, because we've been talking about how free radicals are actually harmful to us, and the same, they are actually harmful to plant cell growth as well. So oxidative stress is just one of the things that plants get stressed about. What are the things that could actually stress out a plant? Wait, that doesn't look quite right. Okay, that's more like it of a stress plant. So heat, virus, fungi, bacteria, free radicals, no water, light, maybe too strong, maybe too weak, and insects are just a few of these reasons why a plant can get stressed. And based on my experience in the lab, although it's just a short 3 years, you can actually see like certain effects on them. Sometimes it's just too late because the plants would sometimes die off, or actually, you do see bruises on the surface of these plants, which makes them not so ideal for experiments, or even use this line for further research. So the reason why we need to understand the messages is we hope to actually capture that they are talking about the stresses that they are undergoing before they actually manifest as physical signs, which means it's too late for us to even intervene and do anything about it because they are probably just too stressed out. So for the last project that we look at the modifications on cytosine, which is the alphabet C, I'm currently still looking at the remaining data generated by the previous PhD student to see what more that we can understand from that. But on the side of it, in my PhD, I get to study a little bit more about the messenger itself, because our previous work in the lab showed that the message, the punctuations on the message are utterly important, but why these punctuations exist beyond the message, and why are they on the messenger itself? Yes, we do know that it's important to kind of have that ID cross-check to make sure that, okay, I'm reading this right. But is there any more importance beyond just that? So in my PhD, I've done a case study on two other modifications that exist on the base adenine, or alphabet A, and the base guanine, alphabet G. So if you remember this slide that I've shown you earlier, I'm going to add two more new elements to it. A message wouldn't just appear out of thin air, so as punctuations. To have these messages exist in the cellular context, you need a writer, which is represented by a pencil, and an eraser. Just in case if you write the wrong modification or the wrong sentence, you can actually remove those. And these two do actually exist as enzymes in the plant cell world. So what have I discovered about the two modifications on the base adenine and guanosine, and what does it have to do with, the enzymes that are responsible in writing this modification? I kind of coined this discovery as, using the analogy from the Marvel Cinematic Universe, so Doctor Strange. And the reason is, this specific writer that actually writes the modification methylated, that by adding a methylated group to adenine and guanosine, modifying the context of the synthesis on the messengers itself, which is tRNA, has actually a time stone property. In other words, what you're watching in Doctor Strange is actually real. Time stone does exist, time control does exist. Not in our world, but in the plant world. So let me explain further. If you remember from Doctor Strange, this is a time stone. It's green in color, and this is how it looks like. We'll put that aside, and this is how the time stone in plant actually look like. Let's name the time stone TRN5, and its function is to actually write a methyl group, CH3, onto the alphabet A and G. But at this stage, you might be wondering, it's just a protein. How does it actually change time or even control time? And how do you know if the time is being changed in plants? So what we found out is, if you remove this specific writer from the plant system, the growth slows down and you get later time flowering. That's weird, right? And we look further into understanding why and how it controls like plant growth time and why it slows it down instead. And what we found out was when we are looking at like the data that we have, the genes that were related to time control, like circadian rhythm, has been affected. We're like, wait a second. Why is it affected? As what we know by then, this specific enzyme actually modifies bases. But where is it modifying and how does it affect genes that control time in plants? And what we found out was without this time stone or this specific enzyme, the modifications on the messenger itself, which you can see here, is a clover leaf structure known as tRNA. The position that's marked in red shows that this is originally where the modification exists. But when you lose the time stone, these bases were no longer modified. And by then, you couldn't get proper translation of the transcripts that actually encode for proteins that control time in the plant. And hence, we named this specific enzyme a time stone, because by just changing the punctuations on the messenger itself, you can actually change time in plants. Isn't that scary but fascinating at the same time? Just by changing the context, you can actually change the consequences in plant growth. And before I end my talk, I would like to provide you with a couple of take-home messages from today's Research Insider. So not all information in the DNA encodes for protein, and in fact, most of them encode for non-coding elements, such as elements to ensure modified RNAs are modified to context to facilitate important cellular processes such as growth. And, And RNA modifications ensure messages within the cell gets through efficiently and accurately. So making sure that they are being transmitted or being passed along at the right context. And plants do get stressed like us. Fundamental research is important because it helps provide understanding of what's actually happening, so we can actually come up with better solutions to actually counter challenges such as climate change and its effect on plants, especially important crops. And these are the references for today's talk. And with that, I would like to end the talk by acknowledging my lab members who have been an important asset, as well as guidance to my research journey. And special thanks to the Bioinformatics Hub, which existed until this year, for actually shaping the earlier parts of my bioinformatics analysis. And to my Science Com inspiration and mentors, Nihin and Daisy, for showing me a lot of like creative ways to do science communication. And a special thanks to my sister, who's helping me out with some of the context of this talk. And thanks again to our organizers, MBIOS, 100 Scientists of Malaysia, and also our media partner, Science Media Center Malaysia, and to the organizations, the University of Adelaide, who has been sponsoring my PhD research scholarship, and to AMSI and Australian Society of Plant Scientists, who has been greatly supportive of my research output. Thank you, and I hope you enjoy my talk. And if you'd like to stay in contact, I've provided my Twitter handle here and the link to my blog post, to my blog page, the Data Midas Diary, if you're interested.
All right, thanks, Sabrina. Gosh, yeah, like, oh yeah, pass it on the chat. It's like a storytelling session. You're quite good at presenting. Thanks for your talk. So
yeah, I was actually quite nervous, so I did stumble a bit, but I really hope that the messages goes out there. Like, hopefully you guys enjoyed it.
Yeah, I have a quick question by Tom, actually. So he wants to know, how do you measure the RNA modifications occurring in the plants in the wet lab context?
So we do measure RNA modification. So there's a couple of ways to measure it. So I wouldn't go into too much of like the technical parts of it, but we do mass spectrometry to measure the modification. So most people might be more familiar with proteomics, so using mass spectrometry to measure proteins. But we can also imply the same technique by just modifying it a bit to actually measure these modifications. So that's one way. The other way of doing it is actually using what we call the next generation sequencing technique to identify signatures. So when bases are being modified, they leave footprints. So I enjoy this kind of like this aspect of data mining a lot, because you have to track down where the modification has actually occurred by following the change bases. So you actually have to understand the code fairly well to identify those. And then we use that as an indicator to look at genome-wide data, like which specific position on the RNA that the modification has actually occurred. So that kind of like compensates for the weakness of mass spectrometry, because mass spectrometry only tells you if there's the presence or absence of this modification. But next generation sequencing actually allows you to go down to single position specificity of where the modification is. That's why we are very certain, like this is where exactly the modification has occurred, and that provides like more context on like how is it important that the punctuation or the modification is modified at the right spot.
Yeah, I think just to add on, correct me if I'm wrong. The method on how you know, when you do your next generation sequencing, the way to find out how one base has been methylated or not is to see, you know, some bases when they get methylated, they get read as a different base. Is that
Yes.
right? Yeah. So
they
say,
get misinterpreted, just
yeah.
like how. Just like how, like, if you say a sentence without indicating where the punctuation is, you might kind of like get the wrong meaning of that sentence. So that's the same actually that happens in
Cool.
the cellular world actually. So you just have to find where they misunderstood it.
Yeah. So I think we forgot to put up a poll that Sabrina has for you guys.
It's all right.
It's okay. We could just do it now while I continue asking questions. So
All right, cool.
There's a question from Hakim. So he said that from your PhD title, it sounds very general compared to DNA modifications. Is it because of the difficulty of handling RNA?
I would say part of it, yes. So a lot of the techniques are still currently under development. So I would say we are a fairly young research field, because out of so many, a lot of these modifications has been studied in DNA because of you know, back in the early days, it's much more the technology that exists, it's much more friendly to study DNA that is much more stable than RNA. That's why a lot of it gets like the information itself get lost during the earlier periods, and only some manage to kind of survive through that technology bottleneck period. Yes, I would say RNA is a pain to handle, especially to even get like high quality RNAs to study these modifications. And imagine that's almost like 150 of them, and sometimes we don't even know that if they actually exist on the same stretch of RNA. So yes, that definitely poses a certain difficulty to it, but we are actually working on it.
Wow, that's very complicated. Yeah, they might not exist on the same RNA, which is going to be a problem. Wow. Okay. Thanks. There's a question from Hakim from Instagram. So I think this is a quite a cute question. So he asked, is it true that a stressed plant would produce more fruits?
Oh yes, I did actually saw that question. So I wouldn't say fruits. So it's like, for example, if you put it in context, like if you're stressed, you can't do anything well. So if you let the plants like get too stressed, you get a lot of flowers. No,
really?
not quoting it from a research paper, but from my plants, they do flower more. So heat stress, for example, actually makes them flower faster and they flower more, but you don't necessarily get the best fruits, you know, like you don't get the best quality of seeds, because they were forced to kind of work under pressure. You're kind of pushing them into I need to survive kind of mode. So when things are kind of like dysregulated in such, more doesn't mean the best. So let's hope they're not too stressed. I'm
Quality versus quantity.
I don't want them to be stressed. Yes.
Okay, cool. So a question by Jake. Are there any scavenger mechanisms to remove free oxygen radicals rather than temporary responses by methylation of bases in plants?
That's a very interesting question, actually. So what happens is methylation is kind of like, okay, hinting the cellular system that this is happening. Guys, take action and stuff. And at the same time, it actually buys time for like the cell to kind of bump up like
antioxidants
systems to kind of remove all these free radicals. Yes, the system exists, but we have to kind of come up with a sentence that to tell every component that's responsible for this, like, hey guys, free radicals are here, do your job, come on. Like, if you don't have that and you don't buy them time, they won't respond like as accurately. So you probably get like gene transcription increase to kind of remove free radicals, but they'll be like, okay, we're here, where's the free radical? Don't know. How fast they want us to remove it? Don't know. Where are they targeting? Don't know. Can't find anything. They'll just walk off. But with that modification, they are kind of like, you can kind of think of it as a mark that tells them, if you get the message here, the SOS message is from here. So you target here. Do not go anywhere else. Understand? Like, yes. So yeah, it's kind of like a way that I would like to pitch it, kind of. So it's actually quite interesting studying plants.
Yeah, I like how you're so passionate about RNA modifications. Yeah, cool. So we have a question from Julian from Facebook.
Okay.
So she asked, have you identified any interesting single nucleotide polymorphisms or modifications in the RNA in the rhodopsis that you're working on?
That's like the reason why I sigh is nucleotide polymorphisms.
Yeah.
It sounds like something interesting, like a footprint, but when you're actually looking at billions of data, I mean, like, let's say from a single plant you actually get billions of data, you have to actually tease it out whether that it's just pure single nucleotide polymorphism, or is that actually like the signature that you're looking for. So that's actually a very interesting question. So that's also the main question that gives me the most headache in my PhD. But we do actually build like an algorithm to kind of tease that apart. So in the pipeline that I've constructed, so based on existing softwares, we do actually kind of tease apart what is just like, the actual SNP that exists purely just a variation, and what's actually the signature. So we do have, not for all modifications, but for certain modifications, like the ones that are presented, we do have a fair knowledge which ones are the SNPs so-called that we are looking for. Yeah.
Oh wow, you built an algorithm. Very impressive.
No, not actually. It's kind of like, just kind of like not actually writing from scratch, not the computer science kind of, but you kind of look at what other people do and kind of just borrow their idea, kind of repurpose it a little bit, because there's no point reinventing the wheel. But if there's something that's quite interesting, and if you kind of use it to explore like a fairly new field, I think it's expanding the work of others. That's what I call it. Isn't that science itself? Like, it's expanding like discoveries after discoveries, and that's how we actually get to know more about, the world around us.
Yeah, it is. We have a question for Ayapa, which is quite interesting. So have you looked into long non-coding RNAs for plants? It's a very new and upcoming subject in epigenetics.
Oh my God. Guys, you are asking a little bit too much about my PhD. Yes, the answer, yes. Actually, I thank him as well for giving me a chance to talk more about other stuff that I've been looking at, because I was thinking when I was structuring this talk, I was like, do I want to tell them that I'm actually looking at a lot of stuff? It's okay, I'll just talk about like a few ones that I'm interested in. And the short answer is yes, I'm looking at long non-coding RNAs, and they are also a different challenge. So the thing is, punctuations, it means different things when it's a long, complex sentence and if it's a short sentence. And for my PhD, I look at short sentences, which is small RNAs, medium sentences, which is messenger RNA, so the ones that actually gets translated into protein, and the one that's just being asked, long non-coding RNAs that kind of ties everything together. So different challenges, but I'm still working on it. That's hopefully by the end of the PhD, I'll get to tell you a little bit about what these modifications are doing on long non-coding. So I was like, so far, this is my current state. So, yep.
That's quite cool. Cool. Looking forward to hear about your success. No pressure.
I hope.
Yeah. So there's a question. What's your favorite RNA modification? So other than methylation.
Oh my. Oh, do I have to pick? So far the ones that I'm looking at, so probably is that position specific modification that I just talked about. So it's two different modifications at the same position, controlled by the same time stone enzyme, which to me is like, why do you have one enzyme doing two jobs? Then I think about it. Yeah, we have one PhD student working on multiple projects. I'm pretty sure one enzyme can work on multiple modifications, not a problem. But it's quite interesting to see like how changing the modification at a specific spot, for example, like ending a sentence with a full stop and ending a sentence with an exclamation mark means a complete different thing. And it depends on what sentence or which paragraph of the sentence that gets modified as well. So that's the same, depending on when that modification was being put on at that specific position, you get a complete different, different outcome as well. So the same position, two modifications, we observed quite a lot of bizarre things that happen. So I didn't really have time to like show you the full spectrum of it. But what we also found out is besides slowing down time, that modification change could actually affect like how the plants grow. So rather than they are branching like this, they kind of go like this.
Oh,
and
wow.
we are like, and if you look at the RNA-seq data, it's a complete mess. It's a complete mess. So if you only try to study the RNA-seq data, you just go, what the heck is happening? If you look at the proteomics data, which we do actually look at it, and I actually look at it, and I go, okay, this doesn't make sense. But if you look at the modification side of it and then compare this two aspects, so this is kind of like a method that I've developed to look at it. So we call it the tri-seq approach. You realize that, okay, now everything makes sense. If you change this, they kind of tell like the other components that this is happening. So we need to change direction, maybe change this direction. No, that's not right. Change this direction. But if it's another modification on a different messenger that gets modified, they'll be like, okay, slow down time. Okay, we need to press the brake on this. Guys, we need to think over. We need to kind of like come up with a contingency plan. So yeah, so far that's my two favorite because they are kind of panicky in a sense. Well, I like to put emotions in my research, so they are kind of panicky and kind of like anxious in a way. So I can kind of relate to them because we do have a little bit of anxiety doing PhD. Yeah, so that's my favorite so far. It's like me. I'm anxious. I'm stressed.
Yeah, I think you probably like the exclamation marks one.
Yeah.
There's a follow-up.
That kind of.
Yeah, there's a follow-up question from Jazang. So how do you identify long coding RNAs from your next-gen sequencing data?
I love the questions today. So long non-coding RNAs, they are very challenging to identify because there are two categories of this long non-coding RNA: the ones that we know and the ones that we don't know.
Yeah.
Unlike the human genome, we know most of it. The problem with plants is we don't know half of it. So you kind of have to the one way to do it is like a collaboration with another bioinformatics group, which is my co-supervisors group. We do have an pipeline to kind of predict new long non-coding RNAs. And it starts with functional study of it. So we have to understand what it's actually doing in the system before we go into like the specific message of it. So what modifications are there, what contexts are there, what message are there, what is it telling the other components of the cell about. So I would say one way of doing it, definitely, actually there's multiple ways of doing it. So one way of doing it, next generation sequencing, you kind of look at you kind of impose like a length filter. So you look at reads that are definitely longer than 200. and then pick out those ones that from the database that you know that it's not coding for protein, and those are your candidates of long non-coding RNA. That's the simplest way to say it. Another way to do it is you kind of do that size selection before you sequence it, and then you look through like long read sequencing. So rather than you chop it out into little pieces and then you try to piece it up together later, you just sequence the entire thing. So that 200 base pair, 800 base pair, you just pipe it through Nanopore or like long read sequencing. You get the entire intact sequence. If you don't accidentally share it before you sequence it, and then kind of like try to put it together and say, okay, these are the amount that I can detect when I isolate for range between this and this, and then try to understand the context. Unfortunately, out of the 150 modifications, there's only 2 long read nanopore sequences algorithms that's available for studying RNA modifications. So the computational biologists and bioinformaticians around the globe is actually still working on like writing different algorithms that allow you to like predict, study these modifications on long read sequencing. But at the same time, the computational biologists are also working on improving using like other informations that's coming out of this long read, like maybe long non-coding RNA and other stuff, to even like kind of like make it a more mature technology to begin with. So we still have a long way to go in terms of studying modification of non-coding RNA, but I think it's promising. Like I've been reading some of the comments. Yes, it's definitely promising. Hopefully before I turn 80, I'll get a chance to study it. Yep.
So you're looking to become a principal investigator then?
Well, first I need to graduate.
That's true. You're so close, so close. So there's a question from Julian again. So do you guys use Nanopore?
We do have a mean ion in our lab. I'm trying to convince my supervisor to let me use it.
Oh.
Still in progress, but let's see how it goes.
Oh, cool. So, you mentioned about the methylases and acetylases, the enzymes that make these punctuations. So, Jake asked him, are there sites in the plant genome that are obstructed from being methylated? I assume that he meant like, how tight the DNA is being curled up and how loose it is for these enzymes to make these marks.
So that's actually a very interesting question. So if the sites are being methylated on the DNA, when it gets transcribed, it doesn't get inherited. So it doesn't, like, for example, let's say in the genome, these sites were being methylated, but when it's being transcribed, the system who copies, so the enzymes that are responsible of copying the messages might look at it like, what kind of modification is this? And why they put punctuation here? Not that accurate. I'll just erase, I'll just copy a new sentence. I'll just copy the base, but probably put the punctuation here, probably put the punctuation here. Yeah, that sounds more right.
Okay, sounds like it has a brain.
So it's a very different system. I try not to think that because it's very scary. Yeah. But it's really interesting in a way that how, the modifications on DNA has its own roles of regulating, for example, like what you have mentioned, the chromatin tightness and everything in terms of making, to control kind of like, it's like a gate control, like access, like making sure like what can access the genome information and whatnot. Some enzymes might try to be sneaky and then let me have a peek what's in there, like, no. And that it's a different regulatory mechanism, but when it kind of gets, the information gets transferred onto RNA, it's a complete different world as well. I'm not sure if you have heard of the concept RNA world. Part of it is because of that as well, because they kind of separate what's happening on RNAs from like what's happening in the genome. Like genome it's a universe on its own. RNA is a parallel universe. Yeah. Although they kind of have similar stuff, but it's different in a way. So I kind of think that Marvel Cinematic Universe is real in a certain extent. Since we have found the time stone, I hope we can find others.
Yeah,
I don't know if it's by me, but I found the time stone. So
it's quite interesting how like whether it's being punctuated or not determines like whether the root grows longer or shorter. Like nothing to do with genome at all.
Well, you might accidentally eat your grandma.
Oh my god. I Another question
But
from Jake.
that actually does happen. So sometimes like missed punctuations due to like, certain external stimuli and stuff, they actually do trigger destructive mechanisms inside plants. And this is not only unique to plants, that's something I would like to point out as well. It does happen in other organisms. Like if you try searching for like, if for anybody that interested, you can try searching RNA modification, human, flies, other organisms, even bacteria. You'll find tons of information on them. I think there are actually certain articles that actually make it easier for you to understand what's actually going on in these. And sometimes like some unexplained causes back in the days about like certain diseases and stuff, they actually found out to be related to this punctuation mark. So it's nothing wrong with the message, it's the context of the message that is wrong. So yeah, it kind of sends, sometimes it accidentally sends destruction signal like eat your grandma kind of thing. So
that's too
make
cute.
sure it's punctuated where it should be punctuated. Yeah. It's real, like you can, it's just the way that I like to describe it, because it's relatable. To me, it's relatable. Science isn't something that is like, it's up in the air, only certain people understand it. It should be accessible. It should be understood by, everybody, because it's happening inside us, it's happening around us. That's what science actually is.
Yeah, I do agree.
Mm-hmm.
Another related question from Jake is that, like all these methylases and acetylases, what dictates the recruitment or the specificity of where it methylates or acetylates on the RNA?
That's another tough question. So the thing is, like, I think Jake's question point out something that's very important in the cellular system. That is, the system is dynamic. It doesn't mean that you try to put a punctuation in that place, you'll definitely stay in that place. Sometimes, like, certain system that exists will try to prevent that, because that's kind of like, you can kind of think of it as a proofreading mechanism. So I would say that in short, there are definitely systems that kind of prevents from happening to a certain site. So one of the, like, I'm not sure how much we have actually understood that on RNA, but most of those on RNA are being obstructed because of their secondary structure. So if you remember from one of my from my slides, you constantly see that folding of like that messenger structure. So this is just an example of how RNA, rather than they exist as a single straight line, they kind of curl up and fold into like kind of like a wavy structure, kind of scrunch up a little bit. So sometimes these things, what we call the secondary structure, does actually obstruct, like the writers and the erasers to come in and like add the punctuation right there, like, I can't get it, oh, forget it, or just leave the sentence like that, whatever, Yeah, you can be like that. Like, I can't add anything in because you are like protecting the messages, like, like in exams when they try to prevent like people from peeking into like your paper and stuff, you're like, no, don't write it like that. Yeah, so there is, but so far our understanding is, as far as I know, is just to structural folding, but there might be more out there. So we'll just have to wait and see how this field actually goes actually.
Yeah, interesting. So a question from Hakim. So bioinformatics looks like the future for life science research. So what would you advise someone new? Where would they start to get into bioinformatics?
So if you look at, so my background is actually genetics and biochemistry. So throughout my undergraduate, I learned close to nothing about bioinformatics. And the reason how I actually got to bioinformatics was because one day I decided to crash an PhD crash course for bioinformatics. So I was hiding behind the workshop actually, and I was like, it's okay, no one will notice me, I'll just follow whatever they're doing. And then the instructor kind of came by and said, I've never seen you. Which lab are you from? Which year are you doing your PhD in? I was like, second year undergrad. I decided to crash. I'm so sorry. I didn't know. Like I know it was for a PhD, but I just wanted to learn. I saw this course. It's really interesting, and that's how I actually got started. So you don't have to wait for the perfect moment to start. You don't have to wait like, oh, I need to sign up for a degree. I need a qualification. Like, learning is more of like, if you really is interested in the field, there are a lot of resources out there. Like you can actually sign up like online courses and stuff. Don't think that just because it's like, a short 4-week course, you might not actually get out of it. But if you never start, you'll never get anywhere. So my best recommendation is, if you have resources around you, check out workshops, check out, even though it says it's for certain people, I don't recommend crashing, but I recommend speaking to them and say, hey, I'm really interested. I know this is not open to everybody, but can I join? Do not crash because it's awkward. Online courses, definitely worth it. There's a lot of like free online courses. My go-to are actually Coursera. So you actually get a lot of like really good bioinformaticians teaching like intro to bioinformatics. My favorite one is actually Rafael's. You can actually check out his tutorials and stuff because it's very easy to understand. It's very accessible, and his contents actually give you like the practical insight is, you're learning this, what are you going to apply it on, and this is how you get a feel of how the field actually likes and what you'll actually be looking at if you choose this as your career path. So Coursera, Rafael's course might be a great way to start. Or if you just want to get a feel of like how coding is, you can actually sign up for like some free workshops on DataCamp, because they have these interactive workshops that you can actually do a little bit of hands-on. They'll give you feedback on like, okay, why isn't it working? What was the trouble? How you should actually write it, and some examples. And you'll find that it's just a different way of thinking. So it doesn't matter if you are not tech savvy, you have no background in coding, you have not done any computer science. If I can do it, I think you can, as long as you keep trying. That's the most important bit. Yeah.
I was gonna say, read Sabrina's blog. That's the answer.
Yeah. So I did actually write a blog on how I transition from wet lab to bioinformatics. So you can actually just type my name and say transition from wet lab to bioinformatics. You'll find a blog post that I've actually written on that, on my journey, how I actually kind of gone from wet lab to dry lab. But this year I kind of gone from dry lab to wet lab again. So I might write another one on that, but so far read the one that's from wet lab to dry lab. That's actually quite, I think in my personal journey, it was a steep learning curve, but I would say definitely worth it.
Yeah, definitely rewarding. I tried a bit of bioinformatics and I was like, I should have done a PhD in bioinformatics. Okay, one last question. So what is your...
I was incorporating it as part of your project. I said like you can try incorporating a little bit of that as part of your project. Like you don't have to like sign up straight full on PhD in bioinformatics. So if you have a little bit of wet lab background, right? And if you can actually have a component of it where you prepare the samples up to how you generate and analyze the data, it actually gives you a better idea what to tell a wet lab scientist or a bioinformatician what you're expecting out of data, because you know that whole process of it. So a lot of the problems that come out when like a bioinformatician and a wet lab scientist work together is they do something, they don't know what things they do that will cause troubles to the bioinformatician, and bioinformatician do something, and then wet lab scientist goes at them and like, why does my data look like this? I do not understand what it means. Like, so I would say both have their advantage. You can be specialized and you have your pros and cons. You can be intermediate, you know a little bit of wet lab and dry lab, but definitely use it to your advantage, because who knows when it will be handy,
Yeah, I think the hard part is persuading your PI to spend a bit of money on next-gen sequencing. Okay, so we'll end with one last question. So what is your career aspiration after your PhD?
Why the audience today are asking so many tough and good questions? So career aspirations, I'm hoping to stay in research. Not because everything has been smooth sailing. I admit I cry a lot, I complain a lot, I thought of quitting a lot. But every single time these kind of emotions or thoughts come up, and then I kind of like, okay, I can finally not do any research anymore. A couple of weeks rest, and I'll be like, I actually miss like trying to solve problems, or actually complaining about research and challenges. And so I hope after I finish my PhD, I would continue doing a postdoc, because I would actually like to see it's just more for my curiosity, actually. I just wanted to see what's more out there, especially throughout my PhD. I get to meet different scientists, and they inspired me like in a different way. They are at different career stages, like, you know, ranging from PhD postdocs to professors, emeritus professors. It wasn't their title or their success that actually attracts me, but when they talk about their personal story about how they actually found something or how they actually discovered something, and people didn't believe it at first, they think it was complete nonsense, and how they actually prove that it's actually something vital. And sometimes, you know, it's just changed how things works in different fields, such as the medical field or maybe like agriculture. I kind of feel that I wanted to continue that path, because who knows, today, like what I'm looking at, to other people it might be just, it's just fundamental research, you know, just to satisfy maybe a scientist's curiosity. You will really never know like when all these toolkits will come in handy, because I believe that a better solution will only come from a really good understanding of how things works. So a postdoc is currently a way to go, but first let's hope I graduate, as I said many times in this talk.
You will graduate.
Yes, I will.
Right, so thank you, Sabrina, and thank you for everyone who joined our chat on this Saturday. So our scientist next week is Prakriti from Monash University, Malaysia. She's doing a PhD in neuroscience and cancer biology, and she's looking into a highly infiltrative tumor that migrates and invades brain regions called glioblastoma. So for more updates regarding future research inside the series, like our Facebook page and bios and 100 Scientists of Malaysia, and see you next Saturday.